Hello, and thank you for joining us. I'm Glenn Taylor, Senior Developmental Editor with Current Protocols at John Wiley & Sons Publishers, and I'm delighted to introduce today's webinar titled Increasing Large-Scale Protein Production Using a Novel Supplement Without Affecting Metabolic Profiles. This webinar is being co-sponsored by Current Protocols and Essential Pharmaceuticals. Essential Pharmaceuticals began operation in 2006 with the release of custodial HTK organ preservation solution in the U.S. for organ transport and transplant. They leveraged their expertise at solving complex metabolic problems that impact cell health to develop a novel solution to optimize bioproduction of therapeutic proteins. Today, our speaker, Dr. Adam Elhofy, will discuss bioproduction methodology challenges currently faced in the industry, along with a new option to optimize protein yield. Current Protocols is in its 28th year and is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research protocols available for life scientists worldwide. With 17 titles and over 17,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. The webinar will be recorded and available for viewing in the next few days. We will send you an email with details on how to access the on-demand webinar, along with a customizable certificate of attendance. And so now it's my pleasure to introduce today's speaker. Dr. Adam Elhofy is the Chief Science Officer of Essential Pharmaceuticals and developed the core technology for the S, that's ESS, line of products. He also aided in creating patents around novel uses of these materials. Dr. Elhofy has 14 years of scientific research experience in the areas of immunology, neuroscience, and oncology. He received the Top 5 Trainee Scientist Award from the American Association of Immunologists for his doctoral research. He also has 14 scientific publications in peer-reviewed journals and has played a variety of roles with startup biotech companies ranging from principal investigator to director of corporate development. So let's go ahead and get started with a very warm welcome to you, Dr. Elhofy.
I'd like to thank you for joining us today. Today I'm going to talk about the use of Cell-Esse in increasing monoclonal antibody production. The topics I'd like to cover today are who we are at Essential, what is Cell-Esse, how Cell-Esse is used as an initial supplement and/or as a feed in protein production, and the associated data describing the benefits of Cell-Esse in protein production, and finally invite questions and discussion. For bioprocessing, the goals are simple: proliferation and production. First, let's take a look at the basic energy inputs for cell growth. There are several different types of energy inputs. For example, carb sources, fats, and proteins. The thinking generally is to turn the TCA cycle and generate sources for ATP. The energy will then be used for replication. This approach implies providing energy for de novo synthesis. The primary objective is to produce protein for biotherapeutic use. The building blocks are amino acids, which can be made and/or added exogenously. The principle is to have amino acids present for protein production. There are consequences for using a lot of fuel, like glucose and amino acids, to power the system. If you add too much fuel to the fire, there can be some toxic side effects. Examples of pushing the system to the limits are lactate, ammonia, and decreased pH. In many cases, and in many talks, you heard the mantra, it is all about the media. The media is considered the vehicle to deliver nutrients and control the system. The available levers you have to pull are typically in the media. As mentioned, one strategy to optimize the media is to provide inputs for the TCA cycle. Here is DMEM-F12 as a model of how to look at different inputs. The categories are amino acids, vitamins as cofactors for generation of energy, and salts needed for the cells to function. Interestingly, there are important things missing. At Essential, we chose to completely rethink the way we approach media. So who are we at Essential? We are one of the leaders in transplantation media. Our perfusion media for transportation of organs, called custodial, shares the majority of market share. Custodial HTK is approved in the U.S. as a medical device. In fact, one of the innovative uses of custodial is in arresting the heart while maintaining cardiac function. While we are relatively new in the bioproduction field, we have been selling this media used for transplantation and open heart surgery for almost a decade. And to this day, custodial is a growing product. We are among the top market leaders in both of these areas. With a slightly different perspective on media and organ survival, we were able to re-examine the approach to cell culture media and develop a novel strategy. The result is, in late 2014, Essential launched into the cell culture media market in support of the biotherapeutic market, which includes cell therapeutics, bioproduction, and primary research. We have a suite of products that are all animal component free, synthetic, and chemically defined. The products cover 3 areas: cell growth, cell freezing, and tissue transportation. Essential is a small company with big capabilities. Our corporate office is in Ewing, New Jersey. We have cGMP manufacturing in both the US and Europe with capacity for large-scale production. Essential also maintains 2 US-based distribution sites. Now, let's dive into the first part of the talk by introducing Cell-Esse's benefits as a supplement used to increase protein production. What is Cell-Esse? As I mentioned earlier, Cell-Esse is animal component free and chemically defined. This leads to greater product consistency and consequently bioproduction yield reproducibility. Cell-Esse's differentiating features make it particularly useful in increasing protein yields as a feed. In the data presented today, we partnered with 2 independent CROs and conducted separate experiments. The goal was to investigate how Cell-Esse would perform in previously optimized serum-free systems. We used a clone designed to grow and produce in Sigma serum-free media and a CHO cell line from LifeTech designed for growth in 40 CHO. We looked at standard parameters of proliferation, titer, yield per cell, production, and metabolism. As some foreshadowing, metabolism in many cases has been used as a gauge to optimize media and increase protein production. In this case, we had an interesting finding. The experimental design was as shown on this slide using the shake flask model. In the first set of experiments, Cell-Esse was used as a supplement only at the beginning of cell culture. Cell-Esse was used at the concentration shown here, starting at 0.5% and increasing up to 15%. But we're only discussing the data up to 5% for today's talk. The experiment was designed to investigate all potential outcomes, including if Cell-Esse is beneficial, if it's detrimental, if there is no effect, or if there is a window where Cell-Esse works or works best. We use the outputs of titer, proliferation, and metabolic profile to quantify the possible endpoints. The study was designed to provide direction for further investigation of optimizing protein expression. So how did it work? As a reminder, these systems had been previously optimized by major companies in the bioproduction space based on metabolic output prior to the introduction of Cell-Esse. Let us take a look at the data. Let's look at the most basic output, namely the effect on antibody titer. There was a significant increase in the titer with the use of Cell-Esse at 1%, but not at 0.5% or 2.5%. The titer was measured by HPLC. As you can see on the slide, the improvements were substantial. The increase in titer was not associated with the increase in viable cell density. In fact, there was no significant cell density difference between the growth of 0.5% and 1% Cell-Esse, but there was a decrease in growth with the use of 2.5% and 5% as mentioned previously. If you use the values calculated from the previous slide, you then get percent increased benefit of using Cell-Esse. The percent yield on a per cell basis is close to 40%. When you look at the ratio of titer to viable cell density, it gives a good understanding of the effect of Cell-Esse on yield per cell. At 1%, there was a significant increase in the production per cell. This can have many implications on bioproduction, including efficiency and batch size. but also including efficiency gains beyond just protein yield, such as scheduling and resource requirements. There is another method for calculating protein yield. You can use the integral under the viable cell density curve with respect to protein yield. This method is more conservative than the yield per cell. Even using this method, there is a significant increase in the amount of protein produced by Cell-Esse. Interestingly, using this method, even at 0.5% and 2.5%, there is a modest increase in production. If you then calculate the percent increase in protein production, you can see there is a significant increase in monoclonal antibodies using Cell-Esse at 1%. Another way to think about optimizing the cell production is looking at the byproducts or looking at the usage profile of the different inputs discussed at the beginning. The metabolic profile, as mentioned above, is a tool to improve proliferative capacity in protein production. As we have seen, there is a significant increase in protein production with the use of Cell-Esse at 1%. Further, there is no change in any of the metabolites that were investigated. This slide contains an important representative group of glucose, glutamate, lactate, and ammonia. While there was a significant increase in monoclonal antibody production, there was no effect on any of the metabolites. We can summarize the first study in this chart showing there was a significant increase in protein production with the addition of 1% Cell-Esse. The significant increase in protein production using Cell-Esse was done multiple times at multiple sites. Here is another study from a different production lab using 40 CHO and CHO cells from LifeTech. The results were similar to the first study. Having these positive results when Cell-Esse was used as an initial supplement, we next wanted to investigate how Cell-Esse would work as a feed throughout the production process. Using a similar design, but now varying the feed and keeping the initial supplement fixed, we investigated Cell-Esse percent feeds starting at 0.5%, going up to 5%. In this study, Cell-Esse and the standard feed were used every 2 days until the cells reached 50% viability. Like with the supplement, there was a significant increase in protein production after the addition of Cell-Esse. In the feed case, the benefit was seen with the use of Cell-Esse at 5%. In this assay, the monoclonal antibody was measured using an antibody-specific ELISA. If you quantify the percent increase in yield, you can see there is a 25% increase in monoclonal antibody titer. The increase in titer with cultures fed with Cell-Esse started early and was increased over control throughout the entire experiment. I will finish with a summary and take any questions. Cell-Esse resulted in a significant increase in monoclonal antibody titer when measured by yield per cell or integral under the viable cell density. When Cell-Esse is used as an initial supplement, the increased yield is not associated with any changes in metabolites. The results are robust and were repeated 3 times in triplicates at 2 different sites, with 2 different serum-free systems and 2 different monoclonal antibodies. When Cell-Esse was used as a feed, it also resulted in a significant increase in protein yield. This presentation has focused on a 2-dimensional approach to protein production. This slide shows a 3-dimensional approach, demonstrating the importance of membranes. The endoplasmic reticulum and Golgi are crucial to protein modifications. Proteins are modified on the membranes of these organelles. At Essential Pharmaceuticals, we chose to rethink the approach to cell culture and provide a supplement that supports the cellular membranes. The results are demonstrated with increased protein production when Cell-Esse is used as either an initial supplement or as a feed. Thank you for your time and attention, and I look forward to any comments or questions you may have.
All right, thank you, Dr. Elhofy. So let's go ahead to the question and answer segment. So if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box that's at the bottom of your screen. Let's go ahead and see what questions have come in so far. All right, to start out, how does this product impact downstream purification?
Well, that's a great question. So far, we've done several standard protein A purifications, and to date, we haven't seen any effect on the purification due to adding of Cell-Esse. But there's another aspect to this that I think that's very important. One of the new technologies that's coming up is constant perfusion, and in that case, people are increasing the amount of cell density without necessarily increasing the titers. And so in that case, you're increasing the amount of host cell protein. And so one of the upsides of using the Cell-Esse is you're able to increase productivity per cell. So in that case, there's actually going to be a positive effect because you're going to have more target protein and less off-target proteins like host cell proteins and less debris from the biomass.
All right. And actually, the next question seems somewhat related to that, so this may be some of the same answer. The question says, with titers at 3 grams per liter, it doesn't seem necessary really to improve the titer, but are there other benefits to be gained?
So that's also a great question. So what we've seen in our results so far is that there's an increase in titer that starts early and actually continues on and stays elevated over control. And so what that does is increases flexibility. So if someone takes down their setup at day 14, they now have the choice to take it down at day 12. So you can shorten the time frame, and so there's some benefit to taking things down 2 days early. The other thing is that with a 20 to 30% increase, you could run the setup a little bit longer and then essentially run fewer bioreactors throughout the year. So instead of running 12 runs, 15 runs, 30 runs, whatever it is, you can reduce the amount of total runs you run per year. And so there's several different ways to think about the logistics in that sense. And you're right that the second part of the question, and that is if you can increase the amount of target protein relative to off-target protein and have higher amount of purity, there's a push from the FDA looking at the amount of host cell proteins. I've seen some reports that have shown that there's as many as 100 host cell proteins that are in the purified protein A group set in the monoclonal antibody purification sets. And so if you're able to increase basically the relative ratios of target protein to off-target protein, so that's another benefit. If you're using Cell-Esse and you're able to get your 3 grams per liter with fewer amount of cells, then that's also a good benefit. And so the last thing to consider is protein quality. And so the way that we approach this is basically we have added a lipid supplement, and so lipid supplement affects the membranes. And membranes don't just include the cellular membrane, they also include the endoplasmic reticulum and the Golgi apparatus. And proteins are made inside of a membrane and then post-translationally modified inside of a membrane as well. And so via using Cell-Esse, there's a chance that you'd be able to stabilize the protein quality throughout the run.
Okay. On to the next question then. Says, when you talk about improving bioproduction, are you adding the supplement to classic media or chemically defined production media?
That's an interesting question. So what we've done so far in the bioprocessing field has been testing in chemically defined media. We've been using it in the LifeTech media, like 40-CHO. We've used it in Lonza media, and we used it in SAFC media, and all of those are chemically defined, and we've had a similar benefit across 3 different media platforms and across different clones. But we also have used this in the context of vaccines, and there the media aren't necessarily as well chemically defined as they are in the large-scale CHO bioprocessing or NS0 bioprocessing. And so there are media choices which are not as well defined, but we've also shown benefit in those systems. So it's a complicated question, but we work in both fields. But the tests that I've spoken about today are those that deal with chemically defined media.
Okay. On to the next question then. So the next one is, does this product increase the cell-specific productivity, or does it increase the total productivity, and how does it impact the glycosylation profile?
So I think that it does increase the titer and the productivity per cell. So the titer increases are, it's been a range. It's anywhere from 20 to 30% increase in titer. The productivity on a per cell basis has gone up to 40 to 50% on an increase on a per cell basis. So it increases both the titer and the productivity per cell.
Okay. On to the next question then is there any impact to the metabolic profile?
Yes, and I left off the answer to the glycosylation portion. So we've tested so far, and I'll answer the metabolic question as well. We have tested the glycosylation profile, and so far we haven't seen any differences between the glycosylation pattern. So the quality of the protein so far seems to be equivalent to the innovator protein. As far as the metabolic profile goes, this has actually been an interesting topic that's come up in the last Bioprocessing International Conference, and I've seen some recent papers. So, and when you use Cell-Esse, none of the metabolites that we looked at were changed despite the increase in productivity by 40%. There was a presentation by a group from Pfizer that showed that they were also able to increase titers without affecting metabolic profile. Their trade-off was they increased the amount of cells by 5-fold and increased the amount of protein by 2-fold. So they increased their biomass and non-target protein significantly without increasing titer significantly. There was another group from Amgen that recently published that looking at the metabolic titers, that basically the readouts obfuscated some of the end products. So if the goal is to look at protein production and not necessarily look at proliferation and cell health, the group from Amgen showed that the lactate profile and glucose profile, that there was basically not necessarily a correlation to protein production between high lactate use and low glucose use. They were looking at something where there was a high glucose use and low lactate, which is inverse in which you would expect to highly proliferating cells, and then that basically increased, there was an increase in the amount of protein. So two different groups showing that there wasn't a significant change in the metabolic profile, but yet there was a significant increase in titer. So our results are consistent with basically showing that you can have a significant increase in productivity without altering the metabolic profile. But this is relatively new theory that people are postulating out there, and so I think our data support that concept.
All right. Moving on to the next question then. The person asks, can I use Cell-Esse with SF9 insect cell culture?
It's a great question. We've been asked that a lot, and so we haven't tested that yet. And so we think that there's a benefit to having lipid supplements in the SF9, but we don't have any data on that one way or the other to date.
Okay. The next question we have here asks, most lipid supplements stick to culture vessels. Does your supplement stick to the vessel or to the tissue culture plastic?
It does not. So there was a paper that was published, it was in 2007, that showed the current status in the lipid supplement field until ours was introduced basically didn't exist. There was no new innovations. And so people either solubilize the lipids in ethanol or they solubilize it in cyclodextran. And this paper that was published in 2007 showed, using isotope tracing, that using either one of those types of modalities to add lipids to your cell culture resulted in most of the lipids sticking to the plastic within 24 hours. After 72 hours, more than 95% of all the lipids adhere to the plastic. The reason that this is important is that most people are moving towards single-use systems. And so by moving things towards single-use systems, which are either polyethylene or EVA, and then if people want to add lipid supplements, the current lipid supplements basically will all stick, and some actually are toxic. And so there's a classic story that we get asked about where someone added a lipid supplement to a wave bag and actually started killing the cells. And that was when the lipid supplement was solubilized in cyclodextran. And those created a sink for lipids and cholesterol and sucked away the lipids and cholesterol from the cells, causing toxicity. Our media does not do that. We tested ours in an EVA wave bag and showed that there was a 30% increase in titer over the control. So not only do we not stick to the plastic, but we still have a beneficial effect.
All right, that sounds great. It is actually now time to conclude the question and answer session. So I'd like to tell you that today's webinar has been recorded and it will be available for viewing in the next few days. We will send you an email with details on how to access the recorded webinar along with instructions on how to personalize and print a certificate of attendance. So on behalf of today's speaker, Dr. Adam Elhofy, and our sponsor, Essential Pharmaceuticals, we appreciate your attending today's webinar and look forward to your attendance at future events from Current Protocols.